Hi, this is the final lecture for microbial metabolism, where we're going to really connect it to the aiptasia microbiome. We're not going to cover all the different types of metabolism for all the different types of macronutrients that life can use. We're just going to cover carbon, nitrogen, and sulfur because for the purposes of our lab, those are the ones we're going to manipulate to try to isolate. Start with carbon. We're going to do a brief review of the TCA cycle and the Krebs cycle and all that good stuff. Microbes are made of over 50% carbon, and this is the carbon that we refer to as the carbon source for biomass building. Carbon is the major metabolic pathway in glycolysis, and it's gives you the most bang for the buck when it comes to energy. And remember, when we're talking about metabolism, we're not just talking about energy, but we're also talking about building biomass. It can get a little confusing as like carbon source for biomass or carbon source for as for energy. Glucose metabolism is the major energy currency for plants and heterotrophs. There are three outcomes for glucose. It could undergo glycolysis to generate ATP and release energy. It could be stored to build biomass, and it's the form of polysaccharides or sucrose, or it can end up in the pentose phosphate pathway, where it's oxidized and broken down to make ribose R-phosphate and make nucleic acids and NADPH for reductive biosynthesis. A lot of times when we think of glycolysis, we see this and we have a mild panic and we decide, oh my God, you throw your hands up in the air. It's like, there's no way I can remember that. But if you break it down into chunks, it becomes a lot easier to kind of understand. And the way that I like to think about it is what's coming in and what's coming out and who can use the waste. So glucose is converted to ATP via glycolysis, which is a TCA cycle. That's this cycle. So first it undergoes glycolysis, and then it enters the TCA cycle. So let's look at glycolysis first. We're going to break this down into simpler chunks. Okay, let's break glycolysis down into simpler chunks. But really what's coming in is glucose, and what's leaving is two pyruvate molecules. And along the way, ATP is being released. Glycolysis breaks the glucose into two pyruvate molecules, releasing ATP. Then those two pyruvate molecules at the end of glycolysis are oxidized into acetyl-CoA, which enters the citric acid cycle. If conditions are aerobic, acetyl-CoA enters the citric acid cycle, also known as the TCA cycle, also known as the Krebs cycle. A lot of times those terms are used interchangeably. And this cycle is a series of chemical reactions that release stored energy. And at the end, one glucose is converted into 38 ATP. That's a lot of energy. So pyruvate's fate in aerobic condition is to enter the TCA cycle. But what happens in anaerobic conditions? Do things die? Do they no longer participate in glycolysis or glucose metabolism? Not necessarily, right? We've all worked out, and when we work out, our muscles can enter anaerobic conditions, but they still need energy. And our body and our cells can undergo fermentation to create energy. Now, it's not as efficient as using the TCA/Krebs cycle, but it does work. And one of the byproducts that you get is lactic acid, which is what you start to feel that burn in your muscles when you are doing exercise. Microbes can also do a different type of fermentation, which is how we get beer and wine. This kind of fermentation's byproduct is ethanol. This also occurs in anaerobic or anoxic conditions. Microbes can also convert pyruvate into 2,3-butyl or other mixed acids, and they're extremely versatile. So ATP, we keep talking about ATP, is the major energy currency for all cells. How is it created? It's created in an ATP synthase through the electron transport chain. In glycolysis, reduced NAD, it's also called NADH, and reduced FAD, and FADH2, are used to create a proton gradient. The electron transport chain has 5 complexes, and these 5 complexes are all increasing in electronegativity, pulling electrons across the electron transport chain. These protein complexes are located in the inner membranes of cells, the inner mitochondrial membrane in eukaryotic cells, or just the cell membrane of prokaryotic cells. And these complexes work together to transport electrons and create a flow of protons, ultimately producing ATP. In complex 1, you have NADH dihydrogenase, right? That is this protein that you see here. And this is in glycolysis. This is the way the electron transport chain looks in glycolysis. Now microbes are metabolically very diverse, and these individual complexes can have different proteins in there, and there are going to be genes coding those different proteins. But at the end of the day, each complex is performing the same action. So in complex 1, it's receiving high energy electrons from a molecule called NADH, which is produced during the earlier stages of cellular respiration. And electrons from NADH are passed through a series of protein subunits within complex 1. And as the electrons move through these subunits, they release energy, which is used to pump the protons, which are the H plus ions, from the outside into the inside of this space here. So complex 2 is a succinate dihydrogenase, and in complex 2 receives electrons from a molecule called FADH2. Here it's labeled as reduced FAD. Also, like whenever you hear reduced, think there's probably a hydrogen added. And it receives electrons from there that are not pumped out, which are not pumped out, and they flow through to complex 3. In complex 3, it's the cytochrome B1 complex. It receives electrons from complexes 1 and 2, and it passes them to complex 4. As electrons move through complex 3, they're transferred between different protein molecules called cytochromes. These are definitely some that you see very different in microbes, but at the end of the day, you're just trying to pull electrons through and manage the proton gradient. Complex 3 also pumps protons across the membrane, right? And in complex 4, it's the cytochrome C oxidase. This receives electrons from complex 3 and transfers them to the final electron acceptor. In this case, in glycolysis, it's oxygen. Oxygen is an excellent electron acceptor. It is basically, I like to think about it as a big magnet that's pulling all of these electrons across. And it's also pumping protons across the membrane, and this is going to end up fueling ATP synthase to generate ADP and turn it into ATP. This proton gradient is storing potential energy similar to like water behind a dam. The protons can only flow back outside through this complex, and also which is ATP synthase. And as the protons flow through ATP synthase, it powers the synthesis of ATP, which is the primary energy source and currency of all cells. The final electron acceptor that's basically facilitating a lot of this is oxygen in glucose metabolism, but it doesn't have to be oxygen, right? This is where microbes can get really creative, and you can get really creative, right? You can maybe eliminate like the kinds of metabolites that a microbe needs for glycolysis to kind of force it to use other types of metabolism that you're kind of more interested in. In bacteria, the final electron acceptor can be something like sulfate or different types of nitrogen. And this also lets microbes survive in really diverse environments. In our model organism at our lab, there are many types of carbon sources that the microbiome can use and the host can use to perform glycolysis, but also different types of microbes can also use carbon dioxide or inorganic forms of carbon to grow and live within its tissues. Some examples of these carbon sources are, glucose. There's an algal symbiont that lives within the plant tissues of aiptasia, and this algal symbiont makes glucose through photosynthesis. It also uses mannose, which is another very simple type of sugar that organisms can use for glycolysis. And this mannose is what creates the cell walls of the dinoflagellate. Acetate another type of carbon that bacteria can use and pop it into the TCA cycle. And then there's inorganic forms of carbon like carbon dioxide. Many chemotrophs use this as not only a carbon source, but an energy source too. We've talked a lot about glucose metabolism. Now, I want you to think about like the electron transport chain, and we're not going to go over that again, but just know that microbes can also use different forms of nitrogen as a terminal electron acceptor through the electron transport chain, right? And that gives it different types of abilities to create energy in different types of scenarios. And what you see back here looks really confusing, but this is a KEGG map, and essentially what you see is different types of nitrogen metabolism pathways, and each one of these letters in a box is the code for a gene that is found in bacteria to facilitate, for example, in this case, turning nitrite into ammonia. Right. So after we isolate microbes in this class, you'll be able to poke into this genome and you can tie it back to the different nutrient sources that you used to grow that microbe. Major nitrogen metabolic pathways include nitrogen fixation, nitrogen reduction, ammonification. Yeah. So some microbes have a diverse array of tools that they can use to manipulate nitrogen in many forms to either create energy or to create proteins. Some of these forms are oxidative processes, which are in yellow, or reductive processes, which are in red. We have assimilation, which is a reductive process by which inorganic forms of nitrogen is reduced into organic nitrogen compounds. And this can be used to build amino acids and nucleotides and for cellular growth and production. Only the amount needed by the cell is reduced in these types of processes. Ammonia assimilation occurs when ammonia or the ammonium ion is formed it was formed during the nitrogen fixation process and is incorporated into cellular nitrogen. We have assimilative nitrate reduction, which is right here. And this is performed by chemolithotropes and is using a reduced or partially reduced form of nitrogen as an electron donor to obtain energy. And it's using this in its electron transport chain, right? So this is what I mean, that it's not always oxygen that is pulling the electrons. And then we have denitrification processes, which is where you're reducing nitrogen. And again, when you're thinking about reducing, think removing oxygens, right? We're losing an oxygen here, so that's a reductive process. Here you're gaining a nitrogen, right? And that's an oxidative process. Denitrification uses the reduction of nitrate into gas, and it eventually turns it into gaseous nitrogen compounds such as N2. And denitrifying microbes perform an anaerobic respiration using nitrate as an alternate finer electron acceptor to oxygen. Anammox is another type. It's anaerobic ammonia oxidation, which is performed by marine bacteria. Here we have a different way of breaking down what we were talking about in the previous slide, but just in terms of thinking about ammonia. Bacteria have different metabolic pathways to use nitrogen as energy, and you can see there's abbreviations of the different enzymes that they use to catalyze these different reactions. So this slide is pairing the different types of processes in the nitrogen cycle and the different organisms that can carry out these processes. This might be a useful tool for you guys when you're thinking about what type of bacteria you're trying to isolate later on. So some examples of nitrogen cycling in aiptasia. So again, ammonium is the easiest form of nitrogen that most organisms prefer. And many bacteria are specialists that can use other forms of nitrogen, right? So nitrogen fixing bacteria can take atmospheric nitrogen, so that's nitrogen that's in the air or in the sky or in the water, that is a gas, and it has a triple bond and it's really hard to break. But nitrogen fixing bacteria can take this nitrogen and convert it into ammonia. This is really important to this could be really important to aiptasia because they're found in really oligotrophic marine systems. Oligotrophic means very nutrient poor. You can actually tell when the type of environment that aiptasia comes in, it's very clear water. When you have very clear water, chances are this water does not have a lot of nutrition. And a lot of times bacteria and symbionts within hosts are what are creating nutrition for the macroorganisms that are living in that environment to be able to live and thrive. Um, so there's limited amounts of fixed nitrogen that are available. So this ammonium or ammonia that's created by nitrogen fixing bacteria can be used by the host. It can be used by the algal symbiont, and it can also be used by other nitrifying bacteria to build biomass or as energy. Nitrifying bacteria might also benefit from being associated to the host because the host creates waste products like nitrites and nitrates that it can use as an electron donor for energy. Some examples of bacteria found in corals and anemones that can use different types of nitrogen. So you have Rhodosudomonas here on the left, and these purple non-sulfur bacteria, they can fix nitrogen and carbon dioxide, and they can also photosynthesize. So they prefer these micro environments that are anaerobic within the coral's tissues, and embedded within the mucus, and also within its gut. And corals and anemones share a lot of the similar characteristics, so it's possible that these can also be in the aiptasia. Bacteria from the Rosiobacter clade can use nitrite as an electron donor, and they're often found in corals in aiptasia. Rosiobacters also account for up to 50% of the coral microbiome, like the mucus microbiome. And a really cool thing about these is they can also degrade sulfur molecules like dimethyl sulfopropanoate, which we'll get into more in the sulfur cycle. But they can, that's often referred to as DMSP, and they degrade DMSP into DMS. So all life needs sulfur for certain amino acids, and some microbes can also use different types of sulfur as an electron donor. Sulfur is something that a lot of us don't think about when it comes to sulfur metabolism, but it is something that we all probably notice when we go to the beach or maybe a swampy type environment. You can tell that it's around by the smell, and those types of sulfur compounds are toxic, and they can be toxic to the life that lives around. But there are partnerships that many organisms have within their microbiome that allow to detoxify these toxic sulfurs into non-toxic sulfates, which the organisms do need to survive. Microbes can play with sulfur and move the electrons around through the electron transport chain, and this process is costly and yields very few ATP compared to glycolysis, but it still gives you energy. There is glucose around bacteria. They're going to probably use that as their best source of energy, but they do have options. Here highlighted in yellow are the oxidative sulfur processes and some types of bacteria that can participate that are capable of oxidizing different types of sulfur. These oxidation pathways are taking toxic hydrogen and adding oxygens, oxidizing it, all the way to sulfate. And this is helpful to the host because sulfates can be used for nutrition and they're not as toxic as hydrogen sulfide, which is very toxic. The red here are reductive processes where oxygens are removed and hydrogens are added via reduction all the way to hydrogen sulfide. While this might not seem beneficial because it's creating toxins, sulfur reducers can help maintain the balance of sulfur cycling of nutrients. And then here you have some other different options of bacteria that you might find that are capable of doing these types of metabolism. And these delta proteobacteria and delsulfurvibrio, those you do see quite a bit in delsulfuromonas in the coral microbiome. So this is a KEGG pathway of sulfur metabolism. I know KEGG pathways can seem a little overwhelming, but the cool thing that you can see in a KEGG pathway is that you can see different types of metabolism. So here's how assimilatory sulfate reduction, right? You can see all the enzymes that a microbe can have that can take sulfate and eventually convert it to sulfide, right? This is a reductive process that you're looking at here. Or the SOX system, which is what you see a lot in sulfur oxidizers, where they can take thiosulfate, which is an ingredient that you'll be able to play around with in the lab, and they can use that for energy, and they can also convert it into different types of sulfate. Um, and again, these processes are really costly compared to glycolysis when it comes to creating energy. So if a microbe that has the capability to metabolize sulfur in these ways to create energy, but it also has glucose there, it's probably going to use glucose. So then you're also going to end up getting a lot of other microbes that also use glucose. So if what you're trying to isolate is something that's capable of using thiosulfate and oxidizing it, or reducing sulfite into sulfide, then you might not want to add. Then adding glucose is just going to, give you more weeds, if you will, instead of what you're actually trying to go after. So, in some examples of possible sulfur cycling in the aiptasia microbiome, sulfates are important for all micro and macroorganisms. Eukaryotic organisms like aiptasia and its symbiont algae take up sulfates, and their wastes are other forms of sulfides. Sulfate oxidizers and reducers can use this waste. In bacterial growth media, sodium sulfate is the default traditional media recipe, but you can also use thiosulfate as a source of partially reduced inorganic sulfur. And what's nice about this is it's not very toxic. And cells can oxidize this to gain useful energy, and then you'll be able to capture sulfur oxidizers and sulfur reducers. Most of these types of bacteria also can fix CO2 as their primary source of cell carbon. So that also means that denying them simple forms of organic carbons will reduce the other types of bacteria that you're going to grow, but it's going to allow you to grow more of these types of bacteria. Another sulfur molecule that is in the aiptasia microbiome is dimethylsulfopropanoate. That's a mouthful. We usually call it DMS. And DMS is a type of algal waste product. They are the primary producers of this type of sulfur. This DMS is also DMSP acts also as an antioxidant and an osmolyte in algal cells, and it's possible that aiptasia can also have benefits from this DMSP. But this DMSP can also be used by bacteria, which can catabolize this and turn it into DMS. And one of the byproducts when they catabolize DMSP into DMS is propanoic acetate, which can be used in glycolysis by the host or by other bacteria. This is a kind of a deeper look at DMSP in the aiptasia microbiome. So the algae can make DMSP, and this DMSP will be converted into DMS and propanoic acetate, which the microbe can use itself into glucose metabolism, or it can provide a source of simple sugar that can be taken into the TCA cycle as part of glucose metabolism by the host. But DMS has a lot of anti-climate warming effects. And the primary creator of DMS is marine bacteria that are degrading the DMSP that marine algae are creating. So we mainly focused on carbon, nitrogen, and sulfur because for the purposes of our lab, those are the nutrients that we're going to be manipulating to be able to isolate different types of bacteria. But all organisms, including eukaryotes, need the same essential nutrients. And this is carbon, oxygen, nitrogen, and phosphorus. If you pay attention to the different enzymes, metabolisms, and genes that different organisms have to use these types of macroorganisms and how they use that to create energy and biomass, you can manipulate them to isolate intended organisms. Micronutrients are also very important for all life, and these are used as cofactors for enzymes. And those are vitamins and minerals. And then the other thing that you need to think about is the temperature, salinity, and pH. And again, I'm simplifying this um, for the purposes of this lecture, but we can kind of probably fill a book with each of these different types of nutrient sources that microbes need to grow. And not just microbes, but anything that's alive. So what we've kind of covered is that microorganisms are metabolically very diverse, and they have way more options than eukaryotes. Bacteria can use different types of nitrogen and sulfur as a terminal electron acceptor, but when given the chance, they're going to go for what gives them more bang for the buck, right? And the way that I think about what's going to give me more bang for the buck is I'll look at a redox tower and pair that with the kind of genes, metabolic genes that a microbe that I'm trying to isolate might have. So, for example, a microbe that can perform both glucose metabolism and sulfur reduction is going to go for the glucose metabolism if it has both ingredients. So if we have an environment without glucose, that same microbe will grow by reducing sulfate into hydrogen sulfide, and microbes that solely rely on glucose for glycolysis will not be able to grow. So this is just a summary of three different types of metabolism and nutrient sources that the microbes that live within our microbiome and the model organism that we're studying, aiptasia, might use, and how we can use these different types of microbial metabolism to isolate bacteria of interest. I really hope that that you know that the sky's the the sky is the limit with all of the options that you can have and all the different types of things that you can isolate and what you can kind of discover through its genome. And I wish you all luck in finding something novel and new.